Welcome to part 2 of teaching you everything that you need to know for the MCAT according to the official AAMC Content Outline. If you haven't seen part 1 covering amino acids, proteins, and enzymes yet, be sure to give it a quick watch because I promise you I covered concepts that will show up on your test day. In today's video, we're going to be covering everything you need to know about nucleic acids, from DNA replication all the way into transcription, translation, and the corresponding tests that researchers use to study nucleic acids, like southern blotting, gel electrophoresis, and Sanger sequencing. So let's drag this out no further and get straight into the video. Before we get into the nitty-gritty details on replication, transcription, and translation, we need an overall knowledge of the fundamentals of nucleic acids. So there are two types of nucleic acids, either DNA or RNA. And the main difference is that DNA is a double-stranded molecule, while RNA only has a single strand. The next major difference comes down to the nitrogenous bases used. DNA uses thymine as a nitrogenous base, while RNA uses uracil in thymine's place. The only difference is that uracil lacks this methyl group that's present on thymine, and the reason why that is because it's a little less energy expensive to synthesize uracil because it lacks that methyl group. And since RNA is generally more short-lived, it doesn't particularly matter that it's slightly more unstable. It matters that it's more energy efficient to produce. Another major difference you might miss if it wasn't highlighted in red here, and it comes down to the 2' carbon position. In deoxyribose, which is the sugar for DNA, it lacks the hydroxyl group and just has a hydrogen. For ribose, the sugar for RNA, you have the hydroxyl group, and this hydroxyl group, although it might not seem like much of a difference, actually makes a pretty big difference in the functions of DNA and RNA. This hydroxyl group, recall, has a lone pair of electrons, 2 to be specific, and that makes it particularly reactive. It can participate in side reactions or be a target for bond hydrolysis, which means that it is higher in energy and less stable. So overall, when we're talking about long-term stability for genetic information, the deoxyribose sugar wins because it doesn't contain this hydroxyl group that can lead to further side reactions. And those are all the differences between DNA and RNA. Taking a further look into DNA, you see that they mentioned the 2 single strands are anti-parallel to each other. And to understand what that means, you first need to have a grasp on the naming conventions. So 5' corresponds to the phosphate group present, and 3' corresponds to the hydroxyl group. So when we say we're moving from 5' to 3', we're moving from the phosphate group to the 3' hydroxyl group. So when you go from 5' to 3' in one direction, the opposite complementary strand must go in the opposite direction from 3' to 5', and that's all anti-parallel means. And what that achieves is it allows the 2 strands to form complementary base pairs and hydrogen bonds. Adenine can form hydrogen bonds with thymine, and cytosine can form hydrogen bonds with guanine because of the anti-parallel nature. Next, we're going to cover the difference between nucleotides and nucleosides, and the difference is pretty easy to understand. So when you look at a nucleotide, you have typically 1 to 3 phosphate groups present, most commonly just 1 phosphate group present because nucleotides are the building blocks of large nucleic acids like DNA and RNA. And since DNA and RNA only have 1 phosphate group present, usually a nucleotide only has 1 phosphate group. And then it's going to have a pentose sugar. So in this case, can you tell what the sugar is and what nucleic acid we have as a result of it? If you said ribose, you're correct because at the 2' position, we have the hydroxyl group and not the hydrogen. So this corresponds to an RNA molecule. And then you have the nitrogenous base, which can be A, T, G, C, or U if it's RNA, which in this case it is. For nucleosides, the only difference is that this phosphate group is gone. So you still have the pentose sugar, which can be deoxyribose or ribose, and you still have the nitrogenous base. You just don't have the phosphate group. And nucleosides with an S are essentially precursors to nucleotides. All you do is you add a phosphate group to the nucleoside, and then bam, it becomes a nucleotide capable of attacking another unit to form a large chain nucleic acid. And the MCAT also likes to pay particular attention to a specific bond, and that is the bond between the phosphate and the sugar group. Right here is known as a phosphodiester bond because it looks like an ester bond in the way that it's attached to a carbonyl and an oxygen, but it has a phosphorus as the central atom instead of a carbon, making it a phosphodiester bond. And this is what stabilizes the backbone of DNA. So you can see here, it's listed and highlighted as sugar phosphate backbone. Phosphate, sugar, phosphate, sugar. This phosphodiester bond is a covalent bond that stabilizes the entire backbone of the nucleic acid. So when we're talking about complementary base pairing, we refer to hydrogen bonds. But when we're talking about the backbone of DNA or even RNA, we're referring to sugar phosphate backbone, which is stabilized by covalent phosphodiester bonds. That's an important distinction to make, so make sure you get it down. Moving on, they want you to recognize that adenine and guanine are purines. You can use the mnemonic pure as gold to help you recognize that purines are double-ringed structures. And let's take a further look at some of their structures just so you could get it into your head. You can see here adenine has 2 rings right here and right there, and guanine also has 2 rings right here and right there. Whereas thymine and cytosine, on the other hand, are called pyrimidines. They only have 1 ring present. And if we're talking about RNA in particular, you can have uracil, which also has 1 ring and is therefore classified as a pyrimidine. And right here, you kind of see misspell. It should be thymine. Threonine is a polar amino acid. It has no place here. So that is a misspell on this study guide, but just replace it with thymine and you'll be okay. Now let's take a deeper dive into the hydrogen bonds themselves. So you can see here that adenine, which always base pairs with thymine, and vice versa, thymine always base pairs with adenine, forms 2 hydrogen bonds. And that's because it each has 1 hydrogen bond acceptor and 1 hydrogen bond donor. So 1 hydrogen bond donor and 1 hydrogen bond acceptor. So it forms 2 hydrogen bonds total. Guanine always base pairs with cytosine and vice versa. You can see here that it forms 3 hydrogen bonds. And that's because cytosine has 2 hydrogen bond acceptors and 1 donor, and guanine has 1 hydrogen bond acceptor and 2 donors. So because it can form 3 hydrogen bonds, it is further stabilized in comparison to the adenine and thymine base pairs. So it takes more energy to split apart guanine and cytosine because it forms 3 hydrogen bonds instead of 2. And if you didn't know this already, DNA and RNA also is a nucleic acid that transmits genetic information. Hopefully you know that. That's kind of basic rudimentary knowledge, but it always helps to reiterate this big picture information so you remember the overall goal as we keep going through these smaller and smaller subsections and details. Remember, the goal is to transmit genetic information. And with this final subsection, we kind of get into replication-oriented details. First, we talk about denaturation and a common way that that's achieved, which denaturing just refers to disrupting the hydrogen bonds, splitting apart the 2 strands of DNA because you're disrupting the interactions that hold them together with most commonly high temperature. It's going to cause the unwinding of the 2 strands. And even though that they're denatured or separate from one another, they could also be brought back together when the proper conditions are restored. And this is called reannealing. And it's possible because although you break the hydrogen bonds, The sugar phosphate backbone, the phosphodiester bonds are covalent bonds that stay intact, and this is what allows them to reanneal after denaturation. For example, a common technique, PCR, which we're going to do a deep dive into, uses high temperatures to denature the two strands. Because the covalent interactions, the phosphodiester bonds, keep the backbone together, when the proper temperature is restored, the DNA strands can come together. But we could also do what is known as hybridization. And when you denature these DNA strands, the single strands are very high in energy. They're very low in stability, so they'll form a complementary bond with whatever. So you insert a complementary DNA sequence of your own, and the single strand of DNA that has been recently denatured or split apart is going to readily bind with the other single strand and create a hybridized fused DNA molecule that is now double-stranded if there is a significant amount of base pair matching. So with the foundations of nucleic acids out of the way, now we could take a deeper look into DNA replication. And before we do so, before we look at the key players and enzymes, I want to give you a zoomed out kind of brief look at the replication fork and how DNA replication and synthesis all starts. So it all starts with two original strands. So you have two parent strands that are separated. The first one you have in the 5' to 3' direction right about here. And the second strand you have from the 3' to 5' direction up top. And because they're anti-parallel, as aforementioned, they're going to have opposite directionality. So those two original parent strands are split apart by an enzyme called helicase. And what helicase does is it splits apart the parent molecules of DNA by breaking apart the hydrogen bonds that connect the bases of the parent strands together. When you break the original parent strands apart, now the replication fork machinery can come in and synthesize a complementary strand to each of the parent strands of DNA, using all the enzymes, most notably DNA polymerase, to carry out the synthesis. And as the replication fork continues to plow down the DNA molecule, you can see here that ahead of the replication fork, you have the natural coiled structure of DNA. But as the replication fork continues to move down the unwound DNA, what happens is they adopt negative supercoils, which are essentially conformations of high torsional stress and strain. So we have a specific enzyme here, DNA gyrase, and what you need to know is that it is responsible for uncoiling the DNA ahead of the replication fork. It gets it back to its proper conformation of low torsional stress so that the replication fork can continue moving down the unwound DNA molecule and continue synthesis. We already talked about helicase splitting apart the DNA by breaking the hydrogen bonds and how that results in the DNA being single-stranded temporarily. And what prevents these two now unwound pieces of DNA from just joining back together and reforming the original DNA molecule, undoing helicase's work, is the single-stranded binding proteins. They stabilize the single-stranded DNA by binding to it to make sure that they don't just reattach and undo the hard work that helicase just did. And with the action of those three enzymes, now we are primed for DNA synthesis. But before we start getting into some of the more key enzymes like DNA polymerase and primase, I want to talk to you about how DNA synthesis actually occurs. And it occurs through a nucleophilic attack from the 3' hydroxyl group. So this is carbon 1, carbon 2, carbon 3 contains a hydroxyl group that's going to attack this electron deficient phosphorus atom, kicking off these extra two phosphate groups and creating a phosphodiester bond. We talked earlier about the phosphodiester bond, and you should know that it is a covalent bond that stabilizes the DNA backbone. And this specific mechanism of attack from the 3' hydroxyl group to the 5' phosphate results in DNA synthesis occurring from the 5' to 3' position. So 5' phosphate, 3' hydroxyl group, 5' phosphate, 3' hydroxyl group, and so on. And this is why we say that DNA synthesis only occurs from the 5' to 3' end, because it requires a free 3' hydroxyl group attacking the 5' phosphate group. And that is how these super long chains of DNA are formed. Just repeated attacks from the 3' hydroxyl group to the 5' phosphate, creating a long, extensive sugar phosphate backbone. But something to note is that we need the 3' hydroxyl group, but it doesn't just come out of nowhere. We specifically rely on the action of primase to lay down a short RNA primer, which provides the free 3' hydroxyl group. So you can't just have DNA polymerase come in and start synthesizing DNA. That's because DNA synthesis does not occur de novo. Essentially, you need a free 3' hydroxyl group, and primase is what creates that free 3' hydroxyl group by laying down a short RNA primer sequence. And once you have this short sequence, then DNA polymerase can take over and make the DNA that is complementary to the original parent strand of DNA. If I lost you or that's a little too convoluted, just remember, you need a free 3' hydroxyl group so DNA synthesis can occur from 5' to the 3' end. And primase achieves that by laying down a short RNA primer that contains the 3' hydroxyl group. With these 3' hydroxyl groups, DNA polymerase can take over and start synthesis by creating a complementary strand to the parent strand of DNA. Now, because DNA synthesis occurs from the 5' to 3' end, we actually get what is known as a leading strand and a lagging strand. So I want you to recall the directionality of the parent strand molecules, 5' to 3' direction. That is the direction that DNA synthesis occurs. So as you can see, it's going in the same direction as the replication fork and its machinery. But if you look at the other anti-parallel strand, you see that the 5' to 3' direction is actually opposite to the replication fork. So what happens here is we create what is known as the lagging strand. And essentially, you get short little bursts of DNA synthesis known as Okazaki fragments. For each Okazaki fragment, you need RNA primase to lay down that RNA primer sequence so that DNA polymerase can act and synthesize DNA. The key thing to remember is that DNA is synthesized discontinuously by a variant of DNA polymerase called polymerase alpha. So since DNA synthesis must occur in the 5' to 3' direction, one of the strands is going to have to move opposite to the replication fork, which results in discontinuous DNA synthesis by DNA polymerase alpha. It requires primase to act on each fragment and lay down an RNA primer so that DNA polymerase alpha can synthesize short DNA fragments that are later all connected by DNA ligase. So each Okazaki fragment requires primase to lay down an RNA primer and ligase to join them all together. And later at the end, the RNA primers that are all laid down are replaced with DNA by a special DNA polymerase. And as you can see, it reiterates the point I made that Okazaki fragments are stitched together by DNA ligase. And for now, we're actually going to table this next bullet point that DNA synthesis is bidirectional. We're going to get into it in a few minutes when we're more down here in this section. But some important points to note is, once again, that biological DNA synthesis always proceeds from 5' to 3' end. And a couple new points that DNA polymerase has proofreading activity. Remember, the DNA that we're replicating turns into mRNA, which turns into proteins. We don't want it to go wrong at any point. There are billions of base pairs within each DNA molecule, and we want to be particularly proactive that we can correct any mismatched base pairs, which is why DNA polymerase has proofreading activity. If it inserts an incorrect base pair, it can recognize and replace it with the proper base pair. And this is essential because if we mess up at the level of DNA replication, then we're also going to mess up at the level of protein production, which in some cases, in some extreme cases, it could be fatal. So we really want to make sure that DNA replication and DNA synthesis is done right, which is why DNA polymerase has proofreading activity. And finally, replication occurs during the S phase. If you don't know much about mitosis or it's a little fuzzy, remember that the G1, S, and G2 phases are pre-mitotic, in which the cell prepares for cell division or mitosis. In the S phase, all that happens is that DNA synthesis occurs to ready the cell to be split into two. So essentially, this process of DNA replication and DNA synthesis occurs during the preparatory S or synthesis, as in DNA synthesis phase. And for the discussion on the semi-conservative nature of replication via the Messelson and Stahl experiment, we're actually going to go to my tablet so I can draw it out and help you better visualize and understand this experiment and how it proved the semi-conservative nature, while disproving the incorrect conservative and dispersive models of DNA replication. So to understand this breakthrough genius experiment, first you have to understand the overall goal. Up until this point, scientists knew that DNA carried genetic information and that it had to replicate to pass that information along. But they didn't know how DNA replication actually occurred. So there were three proposed models that were each tested. The conservative model proposed that the original DNA molecule remained completely intact and was simply copied. The two parental strands stayed together as a double helix, and an entirely new DNA molecule was synthesized alongside of it. So after replication, you would have one completely old DNA molecule and one completely new DNA molecule. The dispersive model suggested that the parental DNA would be broken up into segments, with those parental segments becoming interspersed with newly synthesized DNA. So instead of having entire old and new strands, you would have kind of like a mosaic patchwork of old and newly synthesized DNA distributed throughout the daughter molecule. The third model was the semi-conservative model. This model proposed that the two parental strands actually separated from one another, and then that each parental strand served as a template to then create a new complementary strand. And as a result, each daughter molecule is going to contain one parental strand and one newly synthesized strand. Now we're going to get into the experiment itself. The key to this experiment was finding a way to distinguish old DNA from newly synthesized DNA, and the Messelson and Stahl scientists accomplished this using different isotopes of nitrogen. So a strain of bacteria was grown for multiple generations in media containing N15, or heavy nitrogen, which is represented by the blue DNA. And because nitrogen is incorporated into DNA bases, after several generations, essentially all of the bacterial DNA contained 15N and was therefore relatively heavy. The bacteria were then transferred into media containing only 14N, or light nitrogen, which is represented by the red DNA, and allowed it to replicate. After one round of replication in the 14N media, the DNA was separated according to density using centrifugation, and they observed only one intermediate density band. And this result makes sense under the semi-conservative model, which would propose that the original nitrogen 15 containing strands separate and each one serves as a template for the new nitrogen 14 containing strand. So therefore, every daughter molecule contains one old 15 nitrogen strand and one new 14 nitrogen strand. All of these DNA molecules have an intermediate density, so they produce one intermediate band. Importantly, this eliminates the conservative model. If replication was conservative, the original 15N DNA molecule would have remained completely intact, while an entirely new 14N DNA molecule was produced. We therefore would have expected two bands, one heavy band containing the original 15N DNA, and one light band containing the newly synthesized 14N DNA. Instead, they observed one intermediate band. Then they allowed the bacteria to undergo a second round of replication in the 14N media. And now something interesting happens. The hybrid 15N and 14N DNA molecules replicate. The 15N strand produces another hybrid molecule containing 15N and 14N, while the 14N strand is going to separate and produce a completely light molecule containing of only 14N isotope bands. So after the second round, you're going to have two corresponding unique bands, a 14.5 nitrogen intermediate band and a completely light 14 nitrogen light band. This result now eliminates the dispersive model. Under the dispersive model, old 15N DNA and new 14N DNA would remain interspersed throughout every daughter molecule. Therefore, after each round of replication, you would expect a single band that progressively shifts towards the light position as the proportion of newly synthesized 14N DNA increases. Instead, Messelson and Stahl observed two distinct populations of DNA. Hybrid DNA containing 15N and 14N, and completely light DNA containing only 14N. Therefore, the results supported the semi-conservative model of DNA replication. So with that groundbreaking experiment out of the way, the guide then moves into a discussion of the specific enzymes involved in replication. We talked about all of these at length, except for a few ones like telomerase and nuclease. And telomerase we're going to get into more in depth in a few minutes when we talk about the end replication problem. And nucleases for now essentially help DNA polymerase achieve its proofreading mechanism. So when an incorrect base pair or unwanted or defective segment of nucleotides is identified, nucleases are a class of enzymes that are going to excise or cut out this piece of unwanted or defective DNA. And we also already talked about topoisomerases in our discussion of DNA gyrase, which is a class of topoisomerase. All that topoisomerases do is relieve the torsional strain of DNA when the replication fork pushes through the DNA strand and causes negative supercoiling of the DNA. The topoisomerase introduces a single strand nick in the DNA and relieves the accumulated winding strain. Now we're going to talk about origins of replication. And origins of replications are just the starting point for DNA replication. It's where all the key enzymes and replication machinery meet up to begin the process of DNA synthesis. And the origin of replication starts as something known as a replication bubble. So here we can visualize the replication bubble for a prokaryotic organism. And we know that it's prokaryotic because prokaryotes have circular DNA, whereas we have linear DNA in the form of chromosomes. So this right here is the origin of replication, where replication starts, and it forms bidirectional replication forks. So instead of having to travel a full circle to complete the replication of this circular chromosome, all that happens is it's split up into two forks, and one goes in a semicircle, the other goes in another semicircle, which effectively reduces the amount of time required to copy the entire prokaryotic chromosome. So essentially, we are going to revisit a point that I skipped earlier. DNA synthesis is bidirectional. And what that means is you have two replication forks, and that allows the genome to be copied in a shorter amount of time because now you split up the work between each replication fork, having the time required to copy the chromosome. The difference is that eukaryotes have multiple origins of replication along the same chromosome because our genome is genuinely larger and chromosomes are larger and more dense. So we need multiple origins of replication to ensure efficient replication. So what that looks like is, say you have this chromosome. Over here, you see multiple points of where DNA synthesis is starting. And of course, we also have the bidirectional forks to help save even more times. So what these multiple points of origin achieve is best exemplified by this analogy. Imagine you're trying to copy a 1,000-page book. You have one person that starts on page 1, another that starts on page 250, another that starts on page 500, and another that starts on page 750. Now you could copy that book much, much faster than just one person working alone. And that's what multiple points of origins essentially allow us to achieve. It allows us to replicate our DNA in a much more efficient, much quicker manner. And finally, we're going to move into the discussion of how the very ends of DNA molecules are replicated. So remember in our discussion of the lagging strand, we have Okazaki fragments, and each fragment requires an RNA primer to be laid down to initiate DNA synthesis. After that RNA primer is removed, DNA polymerase can then fill in the DNA that the RNA sequence occupied. But when we get to the very end of the chromosome, and I'm talking the last DNA sequence, after we remove that last RNA primer, DNA polymerase can't fill in that gap because there's no upstream DNA. There's no upstream 3' end hydroxyl group that allows for DNA synthesis to occur. So how do we copy that very last sequence? Well, telomeres are what allow us to copy this sequence. Telomeric sequences are largely repetitive, largely non-coding regions added solely for this purpose to add upstream DNA so that there's a 3' hydroxyl group that the DNA polymerase can utilize to fill in the last part of the chromosome. So essentially, it's kind of junk DNA that is just there for the purpose for DNA polymerase to have some DNA upstream to be able to copy the last part of the chromosome. It doesn't contribute to our genome, but the body knows that this telomeric sequence is essentially non-essential. And what it'll do is it will progressively shorten the telomeric sequence by not copying it every time. And over multiple rounds of DNA replication, eventually the telomeres would decrease so much in size to where they would be gone. And then you would be left in square one where you don't have any upstream DNA again. And that is the end replication problem. Telomeres will progressively shorten until they're gone, if it weren't for the action of the enzyme telomerase. Telomerase has reverse transcriptase activity, so it can use its built-in RNA template to build DNA. And what it does is it essentially replenishes the telomere stores. So rather than allowing the telomere sequence to decrease in length every time, telomerase actually adds DNA sequence repeats to the telomeres to prevent it from getting progressively shorter. So telomeric sequences allow us to copy the very ends of chromosomes. They're our buffer, and telomerases is what replenishes these telomeric sequences. So now that we thoroughly understand the process of DNA replication, let's take a deeper dive, a closer look at the repair mechanisms during and after DNA replication. So I've already mentioned that DNA polymerase has proofreading activity, and that is known as 3' to 5' exonuclease activity. A good way to remember this is that DNA synthesis always occurs from the 5' to the 3' direction. So if we're moving in the reverse direction from the 3' to the 5', this is known as exonuclease activity. And essentially, it just looks and scans for an incorrect nucleotide. And if it finds one, polymerase is going to back up and replace it with the correct one. So proofreading is called 3 to 5 prime exonuclease activity. DNA polymerase also has 5 prime to 3 prime exonuclease activity. And this is essentially what I discussed earlier, the removal of the RNA sequences that are present on each Okazaki fragment or the beginning of the leading strand of DNA and replacing it with the correct DNA sequence. So you want to keep these two separate. 3 to 5 prime exonuclease is proofreading activity, and 5 prime to 3 prime is the removal of the RNA sequence with the correct DNA sequence. And we also have repair of mutations after the process of DNA replication is complete. And you might be wondering to yourself, I earlier said that DNA polymerase is very, very accurate. It doesn't make frequent mistakes. And it also has a proofreading mechanism in place to identify any potential mistakes it might make. So how can it be that there might be mutations after replication is complete given these two factors? Well, that's a valid question, but when we're looking at replication of chromosomes of the genome, we're operating on a scale of hundreds of millions, billions of base pairs. So it is very possible that a few pieces of DNA damage or incorrect base pairs fly under the radar. And that's why we have more mechanisms in place to try and repair those before we go downstream and use that DNA to create mRNA, which creates the protein products. The first kind is called mismatch repair. After DNA replication is complete, it's going to be scanned, the DNA strand, and if a mismatch is detected, then they're going to cut out the whole part of the DNA strand adjacent to the incorrect base pair. And they're going to replace that entire patch that was cut out with the correct nucleotide sequence by DNA polymerase. And then DNA ligase is going to come in and seal that gap. So it's essentially a cut and repair mechanism where you detect a mismatch between base pairs. Like for this one, you see guanine and thymine, they don't form complementary base pairs. So what they're going to do is excise that whole DNA strand adjacent to it, replace it with DNA polymerase, and then seal it with DNA ligase. The next type of DNA repair mechanism deals with a damaged base. So in base excision repairs, sometimes the DNA gets involved in side reactions, and sometimes a nitrogenous base can be alkylated or it can undergo an oxidation reaction, and that is going to change its overall conformation and change the overall shape of DNA. So when a base undergoes these side reactions, what happens is an enzyme called DNA glycosylase is going to find it and bind that DNA and remove that damaged base. And remember, there are 3 parts of a nucleotide. Number 1, you have the phosphate group. Number 2, you have the sugar. And number 3, you have the nitrogenous base. All DNA glycosylase does is it replaces the damaged nitrogenous base. Then AP endonuclease is going to come in and remove that sugar and phosphate. It's going to remove the backbone, which allows DNA polymerase to access the site that was removed and fill in the gap. And then again, DNA ligase seals the nick. The final repair mechanism deals with DNA damage again, and it's called nucleotide excision repair. For this one, I want you to think of one thing always. Nucleotide excision repairs thymine dimers, which are caused by ultraviolet radiation. So whenever you see UV radiation or thymine dimers, I want you to think nucleotide excision repair. And it repairs the helix so it adopts its normal conformation. So UV radiation causes thymine dimers, which is repaired by nucleotide excision repair. And these next 2 concepts are pretty low yield. I don't think I've seen any of them across the 5,000 practice questions I did, but we can go over them regardless in case it shows up in a passage, just so you know the basics. So Nick translation is essentially the name for replacing RNA primers with DNA. So earlier we talked about 5' to 3' exonuclease activity. That's also referred to as Nick translation, in which you replace the RNA primers with DNA. And the SOS response in E. coli is used in emergency scenarios, essentially. So during replication, instead of trying to repair a heavily damaged sequence of DNA, what the SOS repair system does is it overrules the repair mechanisms in place. So instead of correcting any DNA damages, polymerase just uses the damaged DNA as the template and replicates over it, because it essentially operates under the assumption that some DNA is better than no DNA at all. So even though the damaged DNA causes high error rates, some DNA is better than none at all. So it's really used in very emergency situation when you need DNA replication and proteins now. And so that's going to wrap up our discussion of DNA replication. Now, moving down the central dogma, we're going to talk about transcription, which is the process of converting DNA into RNA. So the reason why we do this is because we know that DNA contains genetic information, but the only problem is that ribosomes, which translate the RNA into proteins, are not capable of reading the DNA. They only can read the RNA. So what we have to do is we have to convert the DNA into a readable format. And that's essentially what transcription is all about. So all it consists of is taking the DNA genes and transcribing them into RNA, specifically mRNA. And taking a closer look at this process of transcription, I want to draw your attention to the two most pertinent differences between DNA and RNA. Number one, that RNA is single-stranded while DNA is double-stranded. And number two, that RNA uses the nitrogenous base uracil instead of thymine. So right here we have the original strand of DNA, and the yellow strand is the newly synthesized complementary strand that is going to be transcribed. Off to the right, you have the three names for each of the two strands. And yes, as annoying as it is, you have to know all three names for each of the strands because the MCAT can refer to each of them by any one of these three names. So the coding strand is going to be virtually identical to the mRNA. The only difference is that in place of the T's, we're going to use the uracils for mRNA, whereas the template or non-coding strand is complementary to the mRNA. So this is the strand we use to create our mRNA. It's complementary, which means that it's anti-parallel. So for instance, from the 3' to 5' direction, the sequence TAC corresponds to AUG in the 5' to 3' direction. And that's precisely why these two strands are identical. Again, the only difference being that this has a U, that has a T, because they're both complementary to this strand. So once we have transcribed the DNA into mRNA, the mRNA is brought out of the nucleus and into the cytoplasm to be directed towards the ribosomes to start the process of translation. And essentially, the mRNA is a sequence of nucleotides, as we just saw. The ribosomes reads this nucleotide sequence in triplets. So every 3 nucleotides codes for a specific amino acid. And these 3 nucleotide codes are referred to as codons. Over here, we're going to see a codon chart, which you should see that a nucleotide triplet corresponds to specific amino acids. Now, the anticodons are complementary to the codons, and based off of the anticodon, that's how the tRNA knows which amino acid to bring. So looking at a picture to really understand this better, you have the codon, which is complementary to an anticodon. And based off of this anticodon, the tRNA, which is like a molecular delivery truck, knows which amino acid to attach to and bring to the ribosomes to start polypeptide synthesis. So on one side, the tRNA has an anticodon, which uses the codon sequence to produce a complementary anticodon sequence that then gives the information to tRNA on which amino acid to bring. And to finish out this last section, you see a couple words: degenerate code and wobble base pairing. And wobble base pairing is essentially referring to the flexibility of the third nucleotide position on a codon. So in general, the codon is locked into complementary base pairing with the anticodon. For instance, A would correspond to a nitrogenous base of U because, of course, we're talking about RNA right now, and G would correspond to a base of C. But for the third nucleotide position or the corresponding first position of the anticodon, because they're both complementary, it doesn't necessarily have to follow the typical hydrogen bonding pattern rules. For instance, it could even break some of the typical hydrogen bonds, allowing it greater flexibility. So it makes more sense when we look at a codon chart. And for serine in particular, you see the first two nucleotide positions are UC all throughout. But the third position is a different nucleotide each time: U, C, A, then G. And yet, the anticodon still recognizes the same amino acid for each of the nucleotides in the third position. They all correspond to serine, and that's what makes the genetic code degenerate. So whenever you hear someone or a term of degenerate, that is simply referring to the fact that multiple codons can code for the exact same amino acid. And that's because of the enhanced flexibility of the third nucleotide position in the codon. It does not have to conform or is locked or constrained by typical hydrogen bonding patterns. It can obey atypical base pairing, which allows, again, different codons to code for the same amino acid. And wobble base pairs aren't something that's random. It's actually an evolutionary mechanism that's designed to protect against mutations. So a lot of mutations in the wobble position tend to be silent or degenerate, which essentially means there's no effect on the amino acid. And when we're talking about point mutations in particular, there are three kinds of point mutations: silent, missense, and nonsense. And what a point mutation is when one nucleotide is swapped for another. So there's also mutations called insertion or deletion mutations, which essentially insert or delete a nucleotide, which shifts the reading frame. And we're going to go into more depth later throughout this PDF. But for now, we're going to focus on the point mutations. And in point mutations, all that happens is you change one nucleotide out for the other. For the silent mutation, which is the first type of point mutation, there's going to be no effect on the expression of the amino acid. So let's say we had the sequence ACA and the adenine nitrogenous base was mutated to a guanine. We're still going to code for the same amino acid, threonine, and that's because of the enhanced flexibility of that third position known as the wobble position. So the wobble position essentially protects against potentially harmful mutations, and it leads to degeneracy in the genetic code, where multiple codons can code for the same amino acid. The other two types of point mutations actually have an impact on the amino acid. A missense mutation is when you swap a nucleotide to result in a different amino acid. So again, looking at the codon chart, imagine we have the nucleotide sequence CAU, which would correspond to histidine. But if we change that third residue to and adenine, now we produce glutamine. So instead of producing a histidine amino acid, we now have a glutamine amino acid. So missense mutation is when a single nucleotide change results in a different amino acid. Finally, we have the nonsense mutation, and that results in the production of a premature early stop codon. And we have three different types of stop codons that you are definitely responsible for knowing. Very easy to memorize: UAA, UAG, and UGA. And suppose you have the three nucleotide codon UGU. If that U is mutated to an A, then you get an early premature stop codon. So let's suppose that you are trying to build a polypeptide that is 100 amino acids long. And at the 20th position, the UGU gets mutated into UGA. Now you only have an amino acid or a polypeptide that is 20 amino acids long, missing the other 80. So you could see how a nonsense mutation can definitely have a deleterious or damaging function when a premature stop codon is formed from a mutation. And this section talks a little bit about how initiation is actually started. To start translation, a common thing they'll refer to is the Shine-Dalgarno sequence among prokaryotes. So what this is it's an upstream consensus sequence of the start codon AUG. And AUG is the only start codon, and it refers to methionine. Whenever you see a codon of AUG, you know that translation or polypeptide synthesis begins at that point after until you get to a stop codon. So the Shine-Dalgarno sequence is going to exist a little bit upstream of it, and it's going to help to recruit and align the necessary enzymes or proteins for the ribosome to actually start translation. So before we get to the start codon of AUG, this is essentially the sequence in prokaryotes that helps to organize everything so that translation can properly occur. And we talked already a little bit about the termination codons. Again, you're responsible for knowing each of the three. And we're not going to get too in-depth into the mechanism right now. We're going to get into it a little later throughout this video. But for now, just to orient yourselves, give you a basic overview, a protein called release factor comes along and terminates translation. It terminates translation via a hydrolysis reaction. So whenever it encounters one of these three codons, the release factor is going to come in, perform hydrolysis to end translation. And we finish off this section with a discussion of mRNA. mRNA is very commonly modified after transcription. In fact, it's a necessary process in eukaryotes. Remember, I said that transcription occurs in the nucleus and that translation occurs at the ribosomes, which are contained in the cytoplasm. So the mRNA that is produced in eukaryotes must travel from the nucleus to the cytoplasm in a way that we can increase its stability and decrease the chances that it is degraded by exonucleases is by adding a 5' cap and a 3' poly A tail, and that's going to prevent it from getting degraded. So for eukaryotic mRNA, you must have a 5' cap and a 3' poly A tail. That is a post-transcriptional modification for eukaryotes that allows the mRNA transcript to travel from the nucleus to the ribosomes to begin the process of translation. Prokaryotic mRNAs don't undergo this 5' capping or poly A tail. They don't undergo any post-transcriptional modifications because they don't have a nucleus. For prokaryotes, transcription and translation occurs simultaneously because it's all happening out in the cytoplasm. So as the mRNA is being formed, it's directly being translated by the ribosomes to make proteins. So it doesn't require any form of post-transcriptional modification like 5' capping or 3' polyadenylation that the eukaryotes require. So finishing that discussion of mRNA, the guide then directly moves into a discussion of the other two types of RNA, transfer RNA, known as tRNA, and ribosomal RNA, known as rRNA. And we already talked a little bit about tRNA, and how I described it to you was the molecular delivery truck. So it's going to use its anticodon to bring in the specific corresponding amino acid. And again, that anticodon is complementary to the codon. So ultimately, the codon decides the anticodon, which decides the correct amino acid to bring in during translation. And then rRNA is the catalytic part of the ribosome. It's what actually makes the peptide bond during translation. We already talked about tRNA structure. I showed you it via a picture, but an important thing to note about rRNA is that the large ribosomal subunit is what's responsible for actually catalyzing the peptide bond formation. So when we look at ribosomes in particular, we notice differences between the prokaryotic and eukaryotic ribosome. The prokaryotic ribosome has a 50S large subunit and a 30S small subunit. The eukaryotic ribosome has a 60S large subunit and a 40S small subunit. So for each, the large subunit, respectively the 50S and the 60S, is what's going to carry out the peptide bond formation during translation. And the process of transcription actually starts with initiation, and RNA polymerase is going to bind to the promoter. And in eukaryotes, most frequently, we refer to a Tata box. So this region is very rich in thymine and adenine nitrogenous bases, and it kind of acts as a recognition site for the RNA polymerase to help bind the promoter and start elongation. And the difference between RNA polymerase and DNA polymerase is that RNA polymerase can immediately start adding nucleotides because RNA synthesis can start de novo. So remember when we were talking about DNA synthesis and we mentioned how primase has to come in and lay down an RNA primer for DNA polymerase to work? Well, for RNA polymerase, no primer is required. RNA polymerase can synthesize RNA de novo. So it's going to go down the DNA template and elongate the RNA in the 5' to 3' direction. So for both RNA and DNA synthesis, it's going to be made in the 5' to 3' direction. And now there's two different ways they talk about in which transcription can terminate. The first one is known as intrinsic termination. And as RNA polymerase advances, it encounters a hairpin. And this hairpin is essentially caused by the strong hydrogen bond pairing of guanine and cytosine residues. So as you can see, a GC-rich hairpin loop. And that's because, recall that I mentioned G and C form 3 hydrogen bonds per nitrogenous base. So these very strong interactions allow the base pairs to snap together and form a stem loop structure. And this essentially obstructs RNA polymerase or disrupts it from proceeding, effectively stopping RNA synthesis. So this is intrinsic or row-independent termination. In contrast, we have row-dependent termination, and this is essentially when a protein factor known as Rho chases down the RNA polymerase. So it's also going to move in that 5' to 3' direction, and it's going to catch up to it. And what it does is it has helicase activity. It's going to separate the RNA from the DNA template, which will stop the RNA polymerase from being able to synthesize mRNA using the DNA template. Now we're going to talk about ribozymes. And I remember while studying, I actually kind of found this cool. I don't know if that makes me seem nerdy or not, but it's an RNA molecule that is capable of catalyzing specific chemical reactions. So we see here that RNA can actually have some catalytic activity. It's not only genetic information molecule like DNA is, it actually has active catalysis that it's capable of inducing. And as we move on, we're going to get into a terminology heavy section. So I want to kind of break it down and make it easy for you to understand snRNAs, snRNPs, and spliceosomes. So snRNAs are just small nuclear ribonucleic acids. So they're found in the nucleus. And when they combine with protein complexes and adopt some of that catalytic activity, ribonucleic proteins are going to form together and form a complex known as the spliceosome. And the spliceosome is what carries out alternative splicing. And if you've ever taken a genetics or biochemistry class, you're definitely familiar with alternative splicing. It's extremely important, and it's ultimately how we get a huge variety of proteins through translation. And essentially, it results in one mRNA producing a bunch of different protein products, which are known as isoforms. So if you ever see the term isoform, it essentially refers to different protein products being formed from the same mRNA complex. And this mRNA is going to be filled with exons and introns. Exons are coding regions that are sent to the ribosome for translation, while introns are non-coding regions that are retained. And remember, splicing, exons, introns, all of this is specific to eukaryotes. In prokaryotes, transcription and translation occur simultaneously. But essentially, what splicing is it's a different mechanism of cutting out introns and rejoining exons. There's a bunch of different mechanisms, and that's going to produce a completely different mRNA from what came in. So it also results in a bunch of different proteins being formed. It enables a huge variety of proteins to be formed from the same mRNA. So splicing is extremely important for protein diversity in eukaryotes. And you could see here, for instance, the original mRNA where the introns are just cut out, and then different mechanisms where they join the exons, cut the introns different ways, producing different proteins all throughout from the same mRNA transcript. And we already talked about these post-transcriptional modifications just now and earlier in the video. So if you want, feel free to read over this. It's a nice little handy recap. But otherwise, I'm going to keep chugging along into translation. So we already talked at length about the 3 types of RNA, so I'm not going to go over it again, but feel free to read. It's a really handy summary. And we also talked a little bit about ribosomes and specifically the large subunit. Now we're going to talk about the small subunit. And the small subunit right here is very essential in building the translation initiation complex. So what it's going to do is it's going to gather all the initiation factors, the proteins, and the tRNA, and it's going to use either the Shine-Dalgarno sequence, if we're talking about prokaryotes, or the Kozak sequence to find the mRNA sequence and look for the start codon of AUG to begin translation. So essentially, the small subunit is an essential part of translation initiation. It's what starts the process of building that initial polypeptide chain. And a quick side note that you probably want to pay attention to is FMET, which corresponds to formal methionine. So we talked a lot about the start codon AUG and how that corresponds to the amino acid methionine. Well, in prokaryotes, that methionine is chemically modified into formal methionine. So whenever you see FMET or formal methionine, just know that they're referring to prokaryotic translation initiation. It still corresponds to the start codon of AUG. It just undergoes a little chemical modification to become formal methionine. And I also have a little bit of a beef with the last sentence here, which compares the Shine-Dalgarno sequence to the promoter. So it's essentially saying that what the promoter is to transcription, the Shine-Dalgarno sequence for prokaryotes, of course, is to translation. And although that's a handy heuristic to think about it, it's not necessarily an equivalent analogy. That's because the promoter has a lot of regulation control. The promoter can not only start the process of transcription, but it can upregulate it or downregulate it. Whereas the Shine-Dalgarno sequence, it doesn't upregulate or downregulate or control translation. All it does is it helps position the ribosome to look for the start codon of AUG. It just essentially orients the ribosome and the initiation complex to find the start codon and transition into chain elongation. It doesn't have any regulatory control like the promoter does in transcription, so it's kind of an unequivalent analogy, and I just want you to have all the facts. And so once you find the start codon of AUG, you're going to transition into chain elongation. And that tRNA and the either methionine or formal methionine is going to move in to the P site so that the polypeptide chain can start forming. And essentially, we have three sites when we're talking about translation elongation. The A site, which is essentially the entry site, the P site, where the amino acid chain grows into a polypeptide, and the E site, the exit site, where the fully formed polypeptide chain is going to exit and dissociate from the ribosome subunit. So once we find that AUG corresponding to methionine, that corresponding tRNA and methionine, let's say, is exemplified by this yellow circle, is going to enter the P site, and the mRNA is going to move on to the next codon. And that codon is going to correspond to a specific amino acid, which is going to be brought in by that tRNA. And now the start methionine and the next codon are going to form a peptide bond, which is catalyzed by peptidyl transferase. And that is going to move the corresponding amino acid into the P site. It's going to clear out this A site for the next codon on mRNA to be read, for the next corresponding amino acid to come in and form another bond, growing this amino acid chain. So it's a cyclical process. The next codon comes in that corresponds to a specific amino acid, and that's going to come in with the tRNA, going to form a bond, move to the P site, and grow that amino acid chain until eventually you encounter a stop codon. And you should have the stop codons memorized by now. It's one of three: UAA, UAG, or UGA. And what happens when you encounter that stop codon is GTP is going to be hydrolyzed, and that's going to activate proteins called release factors, and they're going to transition the entire polypeptide chain into the E site. And once they're in this E site, the release factors are going to cleave the fully formed polypeptide chain from the ribosome subunit, and it's going to release the free polypeptide chain. That's going to basically wrap up this process of polypeptide synthesis. So essentially, you find a stop codon, hydrolyze GTP to activate release factors. Release factors are going to cleave the peptide chain, and they're going to exit through the E site. And we're going to go back to the discussion of the initiation complex. So the initiation complex is going to have initiation factors. And to activate those, again, you need GTP hydrolysis. Similar to how you need a GTP hydrolysis to activate the release factors. And to translocate, which means shift. So enter the new tRNA, the new amino acid, and shift the old amino acid into the P site and into the polypeptide chain, you need GTP to activate the elongation factor. So translocation, again, just refers to the transition of the old amino acid to the P site and the new amino acid to the A site. And after this protein is fully formed and dissociated from the ribosome, it does not necessarily mean that it has its full structure and function. In fact, a lot of times we have post-translational modifications that occur in the Golgi apparatus. And via covalent bonds or cleavage of the protein, you're going to alter its activity, its structure, its function, its charge. You're going to alter a lot of it, which can result in activating or inactivating a protein or enhancing its function. So these post-translational modifications, they function to essentially further modify the protein that you get from the ribosomes. So just because the protein is done with the ribosomes doesn't mean it's fully active or in its complete form. And we talked a lot about post-translational modifications in the previous video. If you haven't watched that, I recommend you take a couple minutes, Google phosphorylation, glycosylation, ubiquitination, and learn about how it can activate or inactivate or change the protein's function so that you really understand the importance of post-translational modifications after the protein is fully formed from the ribosome. But other than that, that's going to conclude the process of translation, getting us through the entire central dogma from DNA replication to transcription into mRNA to translation into proteins to post-translational modifications. So let's keep chugging along and dive into eukaryotic chromosome organization. So in general, a lot of times DNA is found associated with proteins known as histones. And what histones are essentially a multimeric polypeptide, which has a positive charge. And it's going to interact tightly with DNA via electrostatic interactions. Because remember, DNA has a very negative charge from its phosphate backbone. So the positive charge of histone proteins and the negative charge of DNA are going to form electrostatic interactions. And this entire complex of histones and the DNA wrapped around it is referred to as a nucleosome. Non-histone proteins refers to all proteins that are not histones. It's kind of intuitive by the name. And mainly they are responsible for carrying out regulatory and enzymatic roles. So everything you see like DNA polymerase or RNA polymerase or even digestive enzymes and proteins, these are non-histone chromosomal proteins. Anything that is not a histone is considered a non-histone chromosomal proteins. Next, we go into the difference between single copy and repetitive DNA. And we've already talked a little bit about a type of repetitive DNA found in telomeres. If you recall earlier in the video, I talked about telomeres and how they contain a highly repetitive sequence of base pairs at the end of chromosomes to prevent the end replication problem, which would cause a progressively shortening chromosome. So I mentioned how they were consistent of repetitive sequences that are not transcribed or translated. They don't have essential coding information. They serve more of a structural capacity. So they have a higher mutation rate because, quite frankly, it's just not as important because it's not genetic information. They exist on a structural capacity. They're not containing the genetic information to create proper proteins or anything. So in turn, there's less error checking and less proofreading for repetitive DNA sequences. Repetitive DNA is also found in structures known as centromeres. And if you don't know what a centromere is, it's essentially the middle portion that kind of holds together the chromosome. So it holds together the sister chromatids. And actually, in mitosis, the sister chromatids are split apart by the centromere during anaphase. And like the telomeres, it's there on a structural capacity. It doesn't contain vital coding information. So in general, it is a highly repetitive sequence that is prone to more mutations because it doesn't contain essential genetic information that is transcribed and translated into proteins. Single copy DNA sequences, on the other hand, constitute most of the organism's genetic information. It is transcribed into mRNA and consequently translated into the proteins. So to ensure that the genetic information produces the correct proteins, we want to make sure that the single copy DNA has a very low mutation rates because we want to produce the correct proteins from the genetic information. And then we already talked a lot about supercoiling and DNA gyrase and topoisomerases. But just to reiterate, during DNA synthesis, ahead of the replication fork, DNA can fold on itself and adopt a conformation of torsional stress. A class of topoisomerases called DNA gyrase is responsible for undoing and alleviating this torsional stress. Now, when we're talking about euchromatin and heterochromatin, you don't necessarily need to memorize this entire paragraph. You just need to understand the difference between euchromatin and heterochromatin. And euchromatin is generally considered to be transcriptionally active. It's classified as loose beads on a string, so it's less dense and associated with active transcription. That's because many times it's not tightly wound up in a nucleosome complex, so it's not very strongly interacting with the histones, so it's more available for transcription machinery to act upon it. On the other hand, heterochromatin is densely packed and largely inaccessible for transcription. You could ignore this point about recent research for MCAT purposes. When you see heterochromatin, that generally means it's densely packed and inaccessible for transcription. And I want you to get away from the thought that DNA either exists in a euchromatin or heterochromatin state. Based on what protein products we need and what genes we need to activate, we can actually convert between euchromatin and heterochromatin. So for instance, if you want to activate a certain gene, it can loosen its association with the histone proteins and be converted into euchromatin. A common way the body achieves this is through acetylation. So it's going to acetylate the lysine groups present on histones, which is going to loosen its interaction with DNA and make it more accessible to transcription machinery. And if, for instance, we don't want to express a gene at a particular point, euchromatin can be converted back into heterochromatin, usually achieved by methylation. And there are some regions that exist only in the heterochromatin state. The two most common examples are telomeres and centromeres, because again, these are acting in a structural capacity. They consist of highly repetitive non-coding sequences. So we want to keep them in that densely packed, inaccessible for transcription state. We don't want to activate them under transcription because they do not carry genetic information. They solve a specific problem or are there on a specific structural capacity. And now we're going to move into some of the high-yield gene expression for prokaryotes. And the way that this is commonly achieved is through operons. And an operon is just a cluster of genes transcribed as a single mRNA. So the genes share a promoter region on the DNA sequence and are transcribed as a whole as a group. The Jacob-Monod model describes the structure of an operon into 4 key components. The first one is the structural gene, and this is going to actually code for the protein we want to produce. The next is the operator site, and this is where the repressor protein, which represses the activity of transcription, will bind. If it's there, it's going to repress transcription. If it doesn't bind here, then transcription is going to be active. The promoter is where the RNA polymerase binds to actually activate transcription. And if the repressor protein is bound, the RNA polymerase cannot bind to start transcription. And the regulator gene is what actually codes for the repressor protein that binds to the operator site when it is needed. Now, genes can be selectively either repressed or positively upregulated in bacteria, depending on the conditions. And the most common example, the most high yield thing you're going to see for operons is the lac operon. The lac operon essentially controls the breakdown of lactose depending on the conditions present in the cell. So right here, you can see the promoter, and this is where the RNA polymerase is going to bind to start transcription of the gene if the conditions allow for it. Right next to it, downstream a little bit, you see the operator, and this is where the repressor protein is going to bind to either block the RNA polymerase, or if it doesn't bind, it allows the RNA polymerase to actively transcribe that gene. And just as we have repressors, we also have promoters, which facilitate RNA polymerase binding. It makes it easier for it to bind. So in times that we want to really upregulate and drive up transcription, we're going to use these promoters to enhance RNA polymerase's activity. And those specific regulatory proteins are known as activators because they're going to activate the transcription and enhance RNA polymerase's ability to bind to the promoter. And that's going to wrap up control of gene expression for prokaryotes. In eukaryotes, we use entirely different methods to control gene expression. And before we get into those, I kind of want to differentiate between the term gene expression and the term transcription. So, the term gene expression refers to converting the genetic information, so for eukaryotes, DNA, into proteins. But transcription, as we know, is the process of converting DNA into mRNA, and that is the first step of gene expression. So, transcription is essentially a facet of gene expression, but the MCAT likes to use these terms more or less interchangeably. So when they say gene expression is upregulated, transcription is upregulated as well. And when they say transcription is upregulated, gene expression is upregulated as well. Just remember that transcription is the first part of gene expression. So technically, they're not the same thing, but the MCAT uses them interchangeably. In the process of transcription, the first step of gene expression actually starts with transcription factors, and these are a class of DNA binding proteins. So what they're going to do is they're going to bind specifically to the DNA. They're going to interact with the grooves in the double helix, and these transcription factors are very essential in recruiting and positioning the RNA polymerase to the promoter sequence. So the RNA polymerase, to start transcription, essentially uses these transcription factors as landmark sites that help them identify where the promoter is and help properly position the RNA polymerase to start the process of transcription. And that's just a general overview of how transcription is started. We can further control transcription or gene expression using enhancers and silencers. And they're pretty intuitive to figure out their function by the names. Enhancers are going to increase transcription, silencers are going to decrease it. And enhancers and silencers both have a couple things in common. Number one, they're both DNA sequences, so they exist along the original strand of DNA. And the other thing is that they can exist either upstream to the left or downstream of the promoter, and they could either be very far from the promoter or very close to it. So their location for enhancers and silencers are not fixed. It's very variable. And to help visualize and get a better understanding of these enhancers, we're actually going to look at a picture. So you can see enhancers pertains to a specific DNA sequence. And what enhancers do is it recruits activator proteins. And once these activator proteins bind, the DNA is going to loop itself over to the promoter, and it's going to form an entire complex with the transcription factors that allow the RNA polymerase to upregulate its activity and to upregulate the levels of transcription. So enhancers are DNA sequences that bind activator proteins, loop over, and help RNA polymerase achieve transcription. On the other hand, silencers are also DNA sequences, and what they're going to do is they're going to recruit repressor proteins instead of activator proteins. And when the repressor proteins bind, same thing is going to happen. That DNA is going to loop and form a complex that prevents the RNA polymerase from accessing the promoter sequence, thereby blocking transcription. So that's the difference between enhancers and silencers. Now, in addition to enhancers, we could also do something known as gene duplication, and that involves just making numerous copies of the gene of interest. So when we need high levels of a specific protein that a gene encodes, what we can do is massively increase or amplify expression by just copying the gene sequence of interest. And usually this can happen in response to specific cellular conditions like hormones, growth factors, steroids, or other emergent intracellular conditions. And the premise of it boils down to, if you have more sequences, more are going to get transcribed, which are thereby going to get translated into proteins, producing more products. So it's very simple. If you have more of a specific gene, there's more of a chance that you produce more protein products corresponding to that specific gene. So that essentially describes gene amplification and duplication. And we already talked extensively about this entire little section here. And I want you to remember that post-transcriptional control only occurs in eukaryotes. Prokaryotes do not undergo post-transcriptional control because they have simultaneous transcription and translation. So the three common ones we went over were RNA splicing, which leads to protein diversity, 5' capping, and 3' poly A tail adenylation. And these two, the 5' capping and 3' polyadenylation, just essentially helps protect the mature mRNA from degradation. So again, when it's being transported from the nucleus to the ribosomes for translation, we have enzymes called exonucleases that tend to degrade RNA. But these specific modifications help it last longer and help it reach the ribosome without actually being degraded. And now we're going to talk about when everything goes wrong. So we've been talking a lot about DNA synthesis and overall a perfect central dogma. But when normal cellular controls fail, sometimes we can have something that we all know and dread: cancer. And before I get into a deep dive on cancer and what it actually is, I want to orient you with the normal growth pathways that we use. So you can see here the RAS pathway, and it's not particularly super important that you memorize the entire pathway. It is pretty lengthy and tedious. You might see it in passages, but the MCAT doesn't explicitly tell you to know every step of this pathway. But what I want you to realize is that there is a lot of regulatory control occurring here before the cell gets the proper signals to proliferate, differentiate, or grow, or divide. So you can see here there's a series of phosphorylations, a series of activations, and a bunch of regulatory proteins here that are all controlling whether the cell gets the signal to proliferate or divide. Essentially, it's a very controlled pathway. But what cancer does is it permanently activates the RAS protein, which permanently gives the cell the go ahead, the green light, the signal to proliferate and divide. So it removes basically all of this regulatory control and just tells the cell, your job is to divide and grow. The cell is getting continuous growth signals. And that is known as an oncogene. And before we have an oncogene, we have a harmless proto-oncogene. So this is a normally functioning, harmless gene that is mutated into an oncogene. And like I said, that oncogene is what speeds up that cell division. It's what sends that continuous signal to the cell that tells it to grow. And it bypasses all the regulatory proteins, usually by inactivating tumor suppressor proteins. So tumor suppressor proteins play a very big role in quality control. They play a very big role at the cell cycle checkpoints, making sure that all the DNA is replicated properly, that there's no genomic instability, that there's no DNA damage. And if there is genomic instability or DNA damage, what tumor suppressor proteins can do is initiate apoptosis. So tumor suppressor proteins play an essential regulatory role that ensures that in a normally functioning cell, growth signals or signals to differentiate or proliferate are only achieved when the normal machinery is functioning properly, when you don't have DNA damage, when you don't have genomic instability. But what happens is that cancer cells are going to mutate and inactivate these tumor suppressor proteins. So now there's essentially no checks, no regulatory proteins on the growth from the oncogenes. So now the cell only gets signals to continue to grow and divide and proliferate without any quality control checks from tumor suppressor proteins. A common analogy to understand this is oncogenes are like slamming down on the accelerator of a car. And tumor suppressor proteins inactivating them is like cutting the brakes from a car. Now your car is stuck in permanent go mode without any brakes or checks on its speed, which is going to lead to uncontrolled cell growth. And we already talked about how cancer cells can avoid apoptosis despite the extensive DNA damage. And the mechanism by which they achieve that, again, is by inactivating tumor suppressor proteins. But they can also stimulate angiogenesis. So now they can get blood vessels to grow to the actively dividing cell, giving it the proper oxygen, the nutrients, the blood that it needs to continue growing at this very quick rate. And here it says that cancer cells are immortal. And while they're technically not immortal, in comparison to normal cells, we talked about the end replication problem, which refers to the fact that telomeric sequences are not infinite. They get progressively shortened, and when they're gone, you can't replicate anymore. This is called a period of senescence. And it's normal to reach senescence as we age, as the cells reach a certain number of finite divisions. And we talked about how telomerase circumvents this by re-upping the telomere sequences. But telomerase is an enzyme, which means it also has finite activity. It doesn't stay around forever, and it's very normal for telomerase activity to decline as we age. But what cancer cells do is they upregulate the activity of telomerase. So now the telomeres are there for good. They're not getting progressively shortened. The cell is no longer going to reach senescence. The telomeric sequence is not going to decrease because the enzyme telomerase is very upregulated. It's going to make sure that the store of telomeres is there for good. And finally, cancer cells can break off and metastasize, which means to grow in another location. And this is what classifies cancer cells as malignant tumors. And then we already talked at length of this entire section about histones, nucleosomes, heterochromatins, and euchromatins, methylation, acetylation, but I'm just going to emphasize the high yield points again because they tend to come up a lot, and I want to make sure you have a good understanding of it. The first, heterochromin, is the transcriptionally inactive, densely packed form of DNA. And when it's in heterochromin, it cannot be transcribed and translated into protein products. But when it's in its less dense transcriptionally active state, it's referred to as euchromatin, and this can be transcribed and translated into protein products. And remember, DNA doesn't either exist in heterochromin or euchromatin. It switches depending upon the cellular needs. We talked a little bit about DNA methylation and how it aids in silencing genes, and you don't need to know the specific mechanism, but just know that DNA methylation generally involves chromatin remodeling from the euchromatin transcriptionally active form into heterochromatin, and that is linked with silencing of gene expression. Acetylation, on the other hand, promotes open DNA, so the euchromatin form, and how it does that is you can see here that the lysine that is found in histone proteins interacts electrostatically with DNA. It forms a tight electrostatic bond with DNA, and they're basically locked together. But when you acetylate this lysine amino acid, you neutralize the charge. So now there's no more electrostatic interactions that is binding up DNA. So now DNA is free from the histone proteins and free to be acted upon. It's no longer locked into a conformation with histone proteins because you get rid of the electrostatic binding that exists between them. And finally, we're going to talk a little bit about non-coding RNAs. And these are functional RNA sequences that are very small, usually about 20 nucleotides in length, give or take a couple of nucleotides. But what they do is they regulate gene expression at the transcriptional and post-transcriptional level. And just a side note, I think it's safe to ignore Pee-wee interacting RNAs. I've never heard of them throughout my biochemistry class, throughout all of my MCAT prep. I don't think they're likely at all to show up on your test day. But the other two major classes are microRNAs and siRNAs. So in specific, microRNAs are these short RNA sequences, and they can act both at the transcriptional and post-transcriptional level. And what they do is they assist in gene silencing. So they're going to bind to a complementary sequence either in the nucleus for the mRNA at the transcriptional level or while the mRNA is being transported to the ribosome at the post-transcriptional level. And when they bind to the complementary sequence, they essentially silence the gene and prevent it from being translated into proteins. So again, microRNA can bind either to the mRNA in the nucleus or as it's being transported. And what it does is it silences the RNA from being translated into the subsequent protein. And this is important because the body wants to achieve homeostatic balance. If we already have enough of a protein product, we don't want to go and make more. So microRNAs are very helpful in silencing that, preventing the RNA from being translated into protein and preventing us from possibly straying from the homeostatic balance. siRNAs are the same exact thing. They can act at the transcriptional and post-transcriptional level, same mechanism, same effect. The only difference is that siRNAs are exogenous. So they either come from viruses or are artificially created by man, whereas microRNAs are endogenous and they exist within your own genome. So that's the only difference between microRNAs and siRNAs. They both have the same mechanism of action, both achieve the same effect, which is silencing, but siRNAs are exogenous, miRNAs are endogenous. All right, the next section we're going to cover goes into biotechnology. It's a little boring, so stay with me. Unless you're a little nerdy and you find this kind of stuff cool, more power to you. Probably help you a lot in your studying. But it starts off with a discussion of gene cloning. And this is very useful for when you identify a particular gene or coding sequence and you want to further study it. Maybe you want to study its regulation or the proteins it produces or how certain mutations can impact its function. It starts with the general same process. You retrieve the gene of interest and insert it into the plasmid. And to do that, first you need a restriction enzyme. And this is what's going to actually help you retrieve that gene of interest. It's going to form cuts around that specific gene and allow you to essentially extract it. And once you have extracted the gene, you want to insert it into a bacterial plasmid. And if you're not familiar with the term plasmid, plasmid is essentially an extraneous piece of DNA found in bacteria. So it lives outside of the main cell chromosome, and it's not a vital part of the genetic information, but it offers things like survival traits. So it can help give traits like drug or toxin defense, or the plasmid can be easily shared. So it doesn't necessarily contain like vital genetic information like the bacterial DNA does. It's a separate component that assists in survivability. And what we want to do is insert the gene of interest from the restriction enzyme into the circular plasmid. And there are three essential parts of the plasmid you're responsible for knowing. The first one is the restriction site, and that's where we're going to actually insert the gene. So we're going to use the restriction enzyme, cut out the original bacterial DNA contained on this plasmid, and insert our gene of interest. The second thing you need is an origin of replication. And we've talked extensively about that during our lesson on DNA synthesis and replication. And essentially, this is where all the replication machinery, the complexes of proteins, the necessary factors for replication assemble to actually start the process of DNA synthesis. So without an origin of replication, you actually can't copy the gene that you're interested. So this is a very, very vital part. You need the origin of replication in each plasmid. Then you need an antibiotic resistance gene, because what you do to study the bacteria is you essentially kill the surrounding bacteria by treating it with an antibiotic. That way you could isolate the gene of interest, the bacteria of interest affected by extraneous factors. It just makes it very easy. So to isolate the bacteria, the plasmid, the gene that you're studying, you need to insert an antibiotic resistance gene. That way, when you kill all the surrounding bacteria, you don't accidentally kill your own plasmid and ruin your own experiment. And then we're going to talk a little bit more about restriction enzymes. So the specific thing you need to know about these restriction enzymes is that to cut and isolate the gene or the DNA sequence of interest, what they do is they cut double-stranded DNA at palindromic sequences. And what palindromic sequences are essentially two complementary strands that read the same exact sequence in the 5' to 3' direction. So for the top strand from the 5' to 3' direction, we have GAATTC. So for the complementary bottom strand from the 5' to 3' direction, we should have the exact same nucleotide sequence. And we do GAATTC. So they both read the same exact sequence in the 5' to 3' direction. And that is what's known as a palindromic sequence. And this is where restriction enzymes actually make their cuts. And they make two different types of cuts, which actually have different effects. So the first type of cut they can make involves an overhang. So it's known as a sticky end, and it creates these little overhangs. And overhangs are very easy to hybridize because these overhangs essentially are not base pairing. They don't form complementary base pairs anymore. They're just free nitrogenous bases. And that's not a very stable conformation for them to exist in. They want to be locked into a complementary base pair. So if you insert a DNA sequence that is complementary to the overhang, they're going to readily snap together and form hybridized DNA. So the sticky end cut creates an overhang that can readily hybridize because it wants to form these complementary base pairs. It doesn't want to exist as a single nitrogenous base. The second type of cut they make does not really often contribute to hybridized DNA, and that's when they cut down the middle and form blunt ends. So as you can see here, there's no overhangs. All the nucleotides are still involved in complementary base pairing. So if you insert a complementary DNA sequence, nothing is going to happen because they're already in a stable conformation of complementary base pairing. So the guideline is going to emphasize that point that sticky ends hybridize, blunt ends do not hybridize. And then we're going to move into DNA libraries, and we will talk about DNA and genomic libraries, but I think it's more important to orient yourself and familiarize yourself with cDNA libraries. So this is going to be the most important bullet point. And the DNA library, much like the name implies, is essentially just a giant book of DNA fragments and DNA sequences that are interesting for further study. Essentially, you cut and isolate specific DNA fragments or genes or coding sequence that you want to further study and put them into a huge database, which is a convenient way of organizing the isolated DNA fragments. Genomic libraries are going to be much larger. They're going to contain more than just coding sequences. They're going to contain introns, promoters, enhancers, silencers, regulatory proteins. They're going to contain it all. So they paint more of a complete picture of transcription. They paint more of a picture than just the DNA fragment. They can tell you the whole story. So for instance, if you want to study the process of how a particular gene creates a particular protein and what happens if you upregulate it, what proteins are involved in the upregulation of it, genomic libraries are going to be much more useful for that particular goal. And the more important definitely is cDNA libraries. And cDNA libraries are made from mature mRNA being reverse transcribed into DNA. So the normal process the central dogma follows is DNA is transcribed into mRNA, which is transcribed into proteins. For cDNA libraries, we have mRNA, which is reverse transcribed into DNA, and then it follows the process of being transcribed into mRNA and translated to proteins. So you might be thinking, what is the point of taking these extra steps where you take a mature mRNA and reverse transcribe it into DNA when we could just get straight to the point, take the DNA and transcribe it like the central dogma normally would? Well, if you identify a protein or a gene in humans and you want to study it via inserting it into bacteria because they could rapidly replicate and allow easy study, you have to make sure that the particular sequence is compatible with prokaryotes. And there are fundamental differences between eukaryotes and prokaryotes that I covered. One is that eukaryotes, like us, we have intron splicing machinery. So our DNA is filled with non-coding sequences known as introns. And when we transcribe that into mRNA, they stay until they undergo the processing of alternative splicing. And that is what's going to remove the introns. And that's what creates mature mRNA. Conversely, prokaryotic organisms do not have introns, and they do not have intron splicing machinery like we do. So if we want to plug in a gene into a bacteria that we found in humans, we have to make sure it doesn't have introns, because the prokaryote, the bacteria, has no way of removing the introns we normally would. So that's why you start off with mature mRNA, because it's already been processed to remove the introns. So you take the mature mRNA lacking introns, and then you convert it to DNA, and then you could go and proceed with the central dogma. The main thing to remember is that cDNA lacks introns because it's made from mature mRNA that has already been processed. In this next section, generation of cDNA, I would just skip over. If you're very hyper vigilant and you want like a 525 and you want to cover every single base, maybe it would be worth it to read over it. But in general, the MCAT doesn't expect you to be a PhD biochemist. They don't expect you to know the entire mechanism of generating cDNA. It's more important to understand what cDNA is and the important points like it doesn't contain introns and the usefulness of cDNA, which it allows us to study human or eukaryotic genes or proteins, instead of just memorizing the entire mechanism that is pretty complex of generating cDNA. So I can confidently say that you can skip this section and get a good understanding of cDNA, and that should be more than sufficient for the MCAT. Then we already talked a little bit about the term hybridization, and this is used interchangeably with the term annealing because they both refer to essentially the same process. So hybridization and annealing both refer to the process of nitrogenous bases in DNA forming complementary base pairs with one another. So whenever you see hybridization or annealing, that just refers to base pairing. And it's a key process of southern blotting, which we're going to talk about in depth, like about 5 minutes from now. And yeah, we already talked about gene cloning, which was essentially the first thing we went over here, and how hybridization can refer the sticky ends on the plasmids where they're not stable in their conformation and are prone to form hydrogen bonds, which of course refers to hybridization. And then we already talked about cDNA, antibiotic resistance, so feel free to skim over this. But we're going to keep chugging along into our first main laboratory technique known as polymerase chain reaction, or PCR. This is a technique you definitely should have heard of and probably have some familiarity with, especially if you're involved in basic science research. It's such a common technique because it's a very, very useful way to amplify your original DNA. So say you have a trace amount of DNA and you want to really amplify it so you have enough DNA to work with to study it. What you're going to do is you're going to use polymerase chain reaction, and that's going to create millions, if not billions, of sequences of the original DNA template. So now you have something to work with instead of just trace fragments of DNA. And it involves 3 essential steps. The first one is denaturation, in which we're going to use extreme heat of 90 degrees Celsius to break apart the hydrogen bonds and separate the 2 double strands of the DNA. And then we're going to cool it down considerably, about on average, 40 degrees Celsius, to allow the primers to anneal with the single-stranded DNA. So they're going to bind to that single-stranded DNA, and you're going to add a lot of primers so they outcompete the reannealing of the template strands. Because we just spent a lot of energy heating it up and separating the double strands. You don't want to cool the reaction and immediately have them stick together. So you're going to import a lot of primers so they essentially outcompete the reannealing. And now During the last step, elongation, you're going to use the DNA polymerase to extend the primers and create that complementary strand. And that is one cycle of PCR, and you're going to keep repeating it to create millions, if not billions, of sequences of the original DNA. And as you can see, it's modeled by an exponential function. So after one cycle, you have 2 copies. After 5 cycles, you have 32 copies. After 100 cycles, you have like billions of copies. I'm not exactly sure what the math works out to, but the point is you get a massive amplification with each successive round from the original DNA template. And now we're going to talk about gel electrophoresis and southern blotting. So gel electrophoresis is commonly used as one of the first steps to actually set up the southern blot test. So gel electrophoresis can be a standalone test on its own, but it's usually used in conjunction with further laboratory tests, just like we're about to discuss with southern blotting. So for MCAT purposes, gel electrophoresis is a good lab technique to separate either proteins or nucleic acids based on their size and their charge. So the charge does affect the separation of the specific molecular fragments, but for MCAT purposes, they like to think of gel electrophoresis as primarily separating on the basis of size instead of charge. So yes, charge does impact migration distances, but unless it's specifically stated in the information or passage, or it's given to you that you must factor in charge, in general, it helps to simplify gel electrophoresis as a separation of proteins and nucleic acids based on size. And the way gel electrophoresis works is you have at the bottom a positively charged anode and at the top a negatively charged cathode. And when you apply an electric field, it's going to make the different molecular fragments travel a different distance based upon their size. So in general, since the nucleic acid DNA is heavily negative charged, you're going to load it at the top at the cathode, and you're going to apply the electric field, which is going to make it migrate to the positively charged anode because opposite charges attract. And the distance it travels is based primarily on its size, and that's because of the gel aspect of gel electrophoresis. So you can use 2 different types of gels, either polyacrylamide if the molecular fragments are smaller, or agarose if the molecular fragments are larger, like, for instance, DNA greater than 500 base pairs. And this gel essentially acts as a molecular sieve. So the smaller fragments can easily slip through it and not get obstructed by it, but the larger fragments are going to have a harder time getting through it and cramming through the gel. So as a result, you're going to see in gel electrophoresis that the smaller base pairs are going to travel much further from the start than the large base pairs. And here you can see the actual results from a gel electrophoresis, and you see a very clean separation of all of the fragments based upon their respective sizes, with the larger base pair fragments appearing closer to the top with a shorter migration distance, and the smaller base pair units appearing closer to the bottom with a larger migration distance. So again, gel electrophoresis just cleanly separates fragments based on their respective size. And like I said, that's often used in conjunction with southern blotting. So once the DNA is separated cleanly by the gel electrophoresis based on their respective sizes, you can then go on to the lab technique southern blotting. And what southern blotting does is it identifies a particular DNA strand of interest. So it doesn't tell you anything about its transcriptional activity, the proteins it produces. It literally just tells you point blank whether a DNA strand is there or not. It just identifies your DNA strand of interest. And the way it does that is, again, after you have this clean separation, what you're going to do is synthesize a radio-labeled DNA probe that is complementary to your DNA of interest, the sequence you want to identify. So you're going to label it with a radioactive phosphorus isotope, and that's going to make it easy to identify. And then you're going to transfer that radiolabeled probe to a sheet of nitrocellulose paper containing the DNA from the gels. And that radiolabeled probe is going to bind to the complementary DNA sequence of interest. And using autoradiography, which exposes its fluorescence, you can easily identify the strand of interest. So to sum it up, to reiterate it, once again, gel electrophoresis separates molecules based on the size. And then using that separation, the southern blot identifies easily whether your DNA strand of interest is present or not. If you want to determine the exact nucleotide sequence of an unknown strand of DNA, that's where you're going to use DNA Sanger sequencing. And in Sanger sequencing, you use three things. The first one is a regular dNTP, which is a regular nucleotide for all intensive purposes, and then DNA polymerase, which synthesizes the complementary strand. But the unique thing about Sanger sequencing is that it uses ddNTPs, and these ddNTPs are missing the 3' hydroxyl group on the proper carbon. So this essentially stalls and terminates replication. So as soon as these ddNTPs are incorporated into the complementary strand, it immediately halts DNA synthesis. And the good thing about these ddNTPs is that they are each fluorescently labeled a different color. So DDA, DDT, DDC, DDG are all labeled with a different color, which allows us to get different colored fragments. So you can see here that we get a bunch of fragments from the gel electrophoresis by incorporating all these ddNTPs that specifically halt the DNA synthesis. And from these different fragments, since they're differently colored, we can easily determine the terminal nucleotide of each fragment. So from this gel electrophoresis, you read from the smallest fragment to the largest one, and we get T, C, G, A, G, T, C, A, G. So now you have the complementary sequence to the original DNA strand. And all you do is determine the reverse complement of this sequence to get the original one. So the key is that you use these ddNTPs, which halt synthesis and are fluorescently labeled a different color to determine the complementary strand. And from this complementary strand, you can therefore determine the sequence of the original DNA strand. Sanger sequencing is essential to determine the exact nucleotide sequence in a strand of DNA. But we also have lab techniques that do more than just tell us whether a specific fragment of DNA is present or the structure of the nucleotides in a strand of DNA. We could actually use some of these lab techniques to analyze gene expression. The first lab technique that we can use to achieve that purpose is known as northern blotting. And the overall goal of northern blotting is to measure the amount of RNA transcripts that are being produced. And from these RNA transcripts, we can therefore come to a conclusion of gene expression. So for instance, if we have a bunch of RNA transcripts measured by the northern blot, we can confidently conclude that gene expression is probably upregulated, because if there are a lot of RNA transcripts being produced, that means that the gene is being actively transcribed into mRNA. And as I said, transcription is often a correlate of gene expression. And then RT-qPCR operates on a very similar principle. And you can recall we already talked extensively about PCR, which essentially just amplifies the original DNA sample. So through the process of denaturing, annealing, and elongation, we get a lot of copies of DNA. And the only difference between RT-qPCR and regular PCR is that this form of PCR starts with an mRNA. So you start with the mature mRNA that is already post-transcriptionally processed, so all the introns are spliced out and everything. And you use an enzyme known as reverse transcriptase to create a complementary piece of DNA known conveniently as cDNA. And remember, we talked a lot about cDNA earlier. And then Now you can use the cDNA as the template for the PCR reaction, because PCR doesn't just directly work on RNA. It needs a starting DNA molecule for it to proceed. And then, based on how many bands are visualized or the density of the bands, we can then determine how much a gene is being expressed. Because if we have a lot of bands that are very dense, we can confidently conclude that gene expression is probably upregulated, because that would mean that we had a lot of RNA transcripts to begin with. And again, if we have a lot of RNA transcripts, similar to the idea and goals of northern blotting, we can confidently conclude that gene expression is upregulated because transcription is actively occurring. Western blotting goes one step downstream of the central dogma, and it operates at the level of translation. So Western blotting, you don't really need to get bogged down in the specifics of how to set it up. You're not a PhD researcher. So the main thing you need to know about Western blotting is it quantifies the amount of protein being produced in the sample. And again, from that amount of protein, you can come to conclusions. So if there's a lot of proteins corresponding to a specific gene that codes for it, we can actively come to the conclusion that the gene is being transcribed and translated and therefore has elevated gene expression. And obviously, none of these three tests are a direct measure of gene expression. We're not measuring directly RNA polymerase activity or the presence of activator proteins or the rate of transcription or translation. We're just using RNA transcripts or proteins as kind of a correlate measure for quantifying gene expression. So if we have a lot of protein or a lot of RNA transcripts, we could come to the conclusion that expression is upregulated because that gene is being actively transcribed into mRNA, and that mRNA is being actively translated into proteins. So it's an indirect measure of gene expression based on the number of RNA transcripts and proteins that we detect in a specific sample. And then they talk about determining gene function. And a common way of doing that is just knocking out the gene and observing the functions or the results of knocking out the gene. So let's say that a group of researchers comes to the hypothesis that a specific gene is linked to increased metabolism. So now what they can do to test that hypothesis is establish two experimental groups. One as the wild type with a functioning gene and one with the gene knocked out. And then if they observe that the knockout gene has decreased metabolism, if they kept all the other experimental variables the same, they could therefore conclude that the gene plays a role in increasing metabolism. So this is a very common way of determining gene function that you're definitely going to see throughout a lot of your basic science passages. We then move into a discussion of stem cells, and we're going to discuss stem cells a lot more when we talk about embryogenesis and the nervous system in the biology section. But for now, I want to orient you with a good overview of totipotent, pluripotent, and multipotent cells. And essentially, they go from least differentiated and least specialized to most differentiated and most specialized. So totipotent cells are the cells present in the very early fertilization structures. So they could be part of the sperm or the oocyte, the morula, the blastocyst. They're essentially the cells that either form the early fertilization structures or the early functioning structures of a fetus. So, for instance, they compose the amniotic sac or the chorion or the placenta. So anything that is very early in embryogenesis is typically totipotent. And these totipotent cells, since they're so low in differentiation and specialization, can become any cell in the body. Next, as you increase the level of specialization and differentiation, as the incoming fetus starts to divide more, you get to the level of pluripotency. And these can become any cell in the body except for the early fertilization structures. So it can't really be the amnion or the placenta or the sperm or the egg, anything like that, but it could be any other cell in the body, excluding the early fertilization structures. And then reach a level of further specialization and differentiation into multipotent cells. And these are the most specialized and the most differentiated, and they're specific to a particular kind of lineage. So they can either exclusively become cardiac tissue or muscle tissue or nervous tissue or respiratory tissue. These cells have already undergone a lot of divisions and a lot of structural and differentiation changes to where now they're being signaled to grow as a specific type of tissue. So these are the most differentiated, specialized type of cells, and these are the least. And then we get into some of the practical applications of DNA technology. And the one that's worth paying attention to is gene therapy. And in gene therapy, this is intended for disease in which a given gene is mutated or has a loss of function or inactivated. So what we do in gene therapy is we essentially take a virus and we're obviously going to inactivate its viral genes and viral proteins and insert a copy of a fully functioning effective gene. And by transferring this normal copy of the genes into the affected tissues where the given gene is mutated or inactive, we hope to restore the function that the gene should have to fix the disease phenotype. So essentially, since viruses are very good at infecting cells and delivering genetic material, we use that to our advantage. We knock out the viral genetic information, the viral proteins, and we insert our own genetic information, which is the functional copy of that gene, in hopes that the virus inserts that information and cures the disease phenotype that results from an inactive gene. And the below paragraphs aren't super important for MCAT content. They're more practical applications. It's not something that they'll test you on your knowledge of. I do want to cover short tandem repeats, which are just short sequences of DNA, like 1 to 6 base pairs, and they're essentially like fingerprints. They are very unique and very specific to a particular person's DNA. And short tandem repeats are a big component of how things like paternity testing is determined. So as humans, we each have our unique 1 to 6 base pair repeat sequence that is found all throughout our genome. And that can be used as a very good way for identification, whether it be through paternity testing or forensics. The idea is that if they sequence those specific STRs, they could identify specific individuals. And you don't really need to know about the agriculture or safety and ethics concerns. They're more of intuitive things, and the MCAT's not going to test your understanding of that. So that's going to wrap up this video where we covered everything on nucleic acids, from DNA replication to transcription to translation to biotechnology. I hope you learned something. I hope you found this video useful. I'll see you in the next video where we cover the next step, genetics. We're going to cover everything that you need to know about the genetics section of the MCAT.